Hello, welcome to part 5 of Biology Paper 1. Okay, so we're going on to straight away to the next question. Duchenne muscular dystrophy is a recessive sex-linked genetic disorder. So that means that it affects males and females more than one than the other. So you may think that colour blindness is like that. It affects more males than females. So figure 14 shows the inheritance of Duchenne muscular dystrophy in a family. State and explain the phenotype of person Z. Okay, so if they're round and they've got like a half moon, they're a carrier female. Okay, so person Z. Oh no, sorry, person Z is a missing one. So I nearly, yeah. So really, this is a different person. Okay, person Z. All right, so we've got a male, female. So they've got 3 children. Okay, 2 females and a male. And this person, who's the missing one, and the female, so it must be a male. So male, first of all. So you've got a male and a female. So that's like these are 2 parents. Okay, yeah, it's really easy to read that wrong because I needed it there. So male, okay. And if so this person's a carrier female. So so you've got unaffected male, affected male. Okay, so it must be unaffected male, so male unaffected. Okay, so we've got again that 25%. Okay, so we've got the heterozygous. Okay, 25%. So he has a male, so he has the dominant allele. Okay. So he has 25% of passing is passing on the genetic disorder. Who needs that dominant allele to have an unaffected daughter. Okay, next question. Blank page 8, completely different thing, not to do with genetics at all now. Potato cells contain the enzyme catalase. This enzyme catalyzes the breakdown of hydrogen peroxide into oxygen and water. So hydrogen peroxide is H2O2, hydrogen and water. Okay. Figure 15 shows what happened when a student placed a potato disc in 5% hydrogen peroxide solution. So hydrogen peroxide solution, test tube, and potato disc. Time. Oxygen bubbles cause potato disc to rise, so that floated up to the surface. The student measured the time taken for the potato disc to rise. The student repeated the investigation using 10%, 15%, and 20% concentrations of hydrogen peroxide solution. Which term describes the hydrogen peroxide in this reaction? Okay, so this is a substrate. It's not the product, it's not being produced. Active site is to do with enzymes. And yeah, it's not control, we're not comparing it to anything. Potato discs all had the same mass. Explain why the student used potato discs with the same mass. So this is the control variable. So mass is a control variable. That's the thing that's kept the same. Okay. So the results can be compared. Okay, right. State 2 other factors that need to be kept the same to 2 other controls during this investigation. So the big one will be temperature, because temperature is a big one in most experiments, does affect most things. So temperature and volume, okay, of hydrogen peroxide. Didn't we talk about that before? All these things are just about experiments again. So we saw that in the other part of the paper, the way that some of it was just about experiments rather than actually knowing anything about the science. So let's have a look. So figure 16 shows the results of this investigation. The student calculated the rate of reaction using 1 over time in seconds to get the rates of concentration, time taken with distillerize, and rate. So 5, 10, 15, 20, time taken. So it just goes down. It's quite a nice, if you drew the graph, it'd be quite nice. Okay, so state and explain a conclusion based on these results. So we've just read with what was happening. So increasing the concentration of hydrogen peroxide increases the reaction rate. So worked out the reaction rate. Okay, this is the reaction rate. So what this explains this. So it explains so scientifically. So higher concentrations. So I need to explain why. So higher concentrations have greater numbers of particles okay, higher concentration, greater number of particles, which increases collisions okay, and breaks down, it's breaking it down. The H2O2, that's the hydrogen peroxide, into oxygen faster. That's the bubbles, wasn't it? That's what we were looking at. We weren't measuring the water. Oxygen bubbles. Okay, that's done. Nice actual question. The student repeated the investigation with the 25% hydrogen peroxide solution and recorded a time of 75 seconds. Calculate the rate of reaction for the 25% hydrogen peroxide solution. Okay, so all we had to do, we've got an equation of 1 over time in seconds. So rate equals 1 over time in seconds. So all we have to do is do 1, just a piece of maths. 1 over 75. Okay, we're going to get a decimal number, 0.13 reoccurring. So 0. Depends how accurate. That was fine. Okay, so we didn't go more accurate in these. Okay, so that's okay. That's be more accurate than that. 0.013 seconds. Just a bit of maths. The student decided that the rate for 25% hydrogen peroxide solution was not anomalous. Give the reason why the result was not anomalous. Okay. So let's have a look what the other actual questions were. So 20 rate. Okay, so it didn't really go up that much, is what that's saying. Okay, so watching everything. It went down a little bit. So it went down a little bit. So that's why I thought it was anomalous. We need to look at the other results for that one. It's not anomalous. So what it is that the enzymes get a limiting factor. So at that point, the substrate, so the concentration, substrate, okay, is not the limiting factor anymore. Not. Because yeah, there is a point you can get to where we're actually increasing the percentage, which concentration will not actually do anything. Okay. Number 9, yeast cells can be genetically modified to produce a painkiller. Okay, this painkiller is usually obtained from opium poppies. So is that codeine? So one method for genetically modifying a yeast cell uses a plasmid containing the desired gene. Explain how a gene can be inserted into a plasmid. This is a bit like when you make insulin, isn't it? So what happens? So the gene in the plasmid, is cut. Okay, that's what it's actually called. Cut using a restriction enzyme. Okay, so this is inserted into the yeast cell. Okay, using ligase. Very similar to when you make insulin. Okay. Discuss the possible benefits of producing painkillers from genetically modified yeast cells rather than extracting the painkillers from poppies. So it's not like when you have to kill animals to get the insulin. It's not a thing to do with that. It's more to the speed. Okay, so yeast grows rapidly. It's more the speed with this one. Yeast grows rapidly. Okay, yeast grows rapidly. That's its benefits. And it takes up less space than growing poppies. Because that's going to field, wouldn't they? Okay. And not weather dependent. Yeah, because poppies, obviously, they're plants that they need certain weather conditions. Okay, risks. Okay, risks. Okay, so GM organisms may contaminate the environment. Okay, so it can enter environment and damage ecosystems. Okay, we've got 3 points there because actually had a choice to do of you won't get any more marks or anything else for that one. Okay, next one. Figure 17 shows the structure of a DNA nucleotide. Okay, so structure A is a sugar. So there's that kind of pentagon shape, sugar. Okay. Right, so we get into the 6-mark question. So the star means that we are going to be marked on our style of writing. So we have to write full sentences for this one. Okay, so there's 1 mark for the comprehensive. So you need to say that comprehension and sort of how it's read. That's what that means. So in 2003, the first complete human genome was sequenced. The genomes of different people have small changes in the sequence of DNA bases. Describe how these changes in DNA sequence can affect the individuals and how sequencing a person's genome could influence their medical treatments. Okay, so DNA has 4 different bases. Okay. Okay, so changes to DNA and mutations. So individuals, so even people, individuals have different alleles. alleles for genes. Okay, so sequencing can identify genetic diseases or the risks or risk factors of diseases. So this can allow treatment of the genetic disease treatment or preventative action. So scroll down a little, it's going to have some more room. Preventative action. So it could be in lifestyle changes. So say, example, diabetes, that people can sequence genes for, and someone could change their lifestyle to prevent the disease either becoming worse or developing. Or preventative medicine, so it could be another medication for the individual. Okay. Yeah, this is done quite a lot now to see what people could develop so they can take action before it happens. Okay. Number 10, a scientist is planning to compare the effectiveness of the antibiotic mixopyronin on 2 different species of bacteria. Figure 18 shows the equipment the scientists can use: agar plates, 2 species of bacteria, filter discs of mixopyronin, so they're actually on its spreader, incubator. Describe how the scientists can determine the effectiveness of mixopyronin on 2 species of bacteria. Okay, so antibiotics, so that's the type of antibiotics. So antibiotics kill bacteria or prevent them from reproducing. Okay, that's a bit of background. Okay, so spread each bacteria type on different agar plates. Different agar plates. Okay, add the mixopyronin discs to each. Incubate the agar plates. Incubate the agar plates and measure the zone of inhibition. So what the bacteria have been killed or not grown around them. Okay. Okay, next one. So mixopyronin inhibits bacterial RNA polymerase. Okay, explain why the antibiotic mixopyronin can be used to treat bacterial infection in humans. So same sort of thing again. So antibiotics, the same thing. Antibiotics, they either kill bacteria or prevent reproduction, prevent bacterial reproduction. So what happens then? So it doesn't affect humans. So it doesn't affect human RNA. So prevents transcription in bacteria, transcription in the bacteria. So prevents protein binding. So that means no protein synthesis. Okay. Okay, 6 mark. Right at the end of the paper now. Okay. So infections can also be caused by viruses. Describe the lytic pathway of a virus and how this causes the spread of infection through a population. So people kind of probably learned a lot about viruses recently. Okay, so this is all about, it sounds more difficult than it is. Okay, so viruses, remember they're like parasites. So viruses cannot replicate, okay, outside a host. So they need something, they need a host. So again, like a parasite. Okay, they use host cells, like so even your own cells, okay, cells to produce viral proteins by inserting genetic machinery into the host cell. Okay, and then the viral proteins assemble, they put themselves together into viral particles. Right, what this does, so these exit through the host cell membrane. Okay, this is called lysis, so it tells cells to burst, causing lysis. Okay, so what this does is the spread. So virus particles leave the host in bodily fluid, so coughing, sneezing, things like that. And pass on to other members of the population by bodily fluid transmission such as coughing and sneezing. etc. Okay, that's the end of that one. So probably all know quite a bit about the virus, but we can also draw a diagram as well to help you out with that one. So I'll leave that on the screen. So that's the end of that paper. Okay, Andrew Mark, well done for making it all the way through. Thank you for listening and hope to see you in another video. Thank you and goodbye.